Assalamualaikum warahmatullahi wabarakatuh and a good day to my lecturers. I am Muhammad Muqri bin Yasimituah, a final year student of Resource Biotechnology, and today I would like to present my research proposal for my final year project entitled Viability of Encapsulated versus Free Cell in Different Environmental Conditions. So what is cell encapsulation? Cell encapsulation refers to cells which have undergone encapsulation. It is a process where the bacteria are artificially coated or entrapped within a known substance, usually a biofilm. There are three methods in producing an encapsulated cell. First, emulsion method, extrusion method, and finally drying method. In my research, I will be using the extrusion method. So what are the uses of encapsulated cells or immobilized cells? It is used in various biotechnological industries. It's used as a biological soil repair. It's used in sewage treatment facilities. And finally, it's also used to produce antibiotic, enzymes, and amino acids in a large scale. For the background of my study, encapsulated cells are prominently used in many biotechnological institutes and have a high demand in the industrial sector. However, not much study has been made about the viability of encapsulated bacteria against harsh environmental conditions that would have an effect on its free-living counterparts. The objectives of my research are, first, to study the viability of encapsulated cells as compared to free-living cells against the same harsh environmental condition. Next is to identify the degree of viability in encapsulated cells against the defined environmental condition. For my literature review, I would like to talk about the E. coli bacteria, immobilized bacteria, and the parameters which I'll be using in my experiment, which are temperature and pH value. So first, for E. coli bacteria is a typical bacterium found in the gastrointestinal tracts of humans and other vertebrates. It is found in 1885 by Theodore Escherichia, where he was studying human feces. In short, E. coli bacteria is a gram-negative, rod-shaped, facultative anaerobic neutrophilic bacterium. Most of the strains are harmless with an exception for few, such as the ED157H7 E. coli strain, which can cause stomach pain and also intestinal problems. So why E. coli? I've chosen E. coli due to a few reasons. First, it is the most commonly used bacteria in both research and industrial purpose. Second, it has an easy thriving condition with an optimal pH of 7 and can be kept at room temperature. Hence, it is easy to be taken care of. Finally, it has a relatively rapid growing rate, saving me time during my research. Even so, not much study has been done regarding encapsulated E. coli bacteria. For immobilized cells, I've researched an article by Zur et al. In 2016. Immobilized cells are founded by Vincenzo Bisceglia in 1934, and he is the first person to encapsulate cells. The first cells encapsulated are tumor cells. It is believed, according to Zur et al., that the threshold of immobilized microbes will be higher in harsh environmental conditions. The most common method of encapsulation is via biofilm, gelling goo, or calcium alginate. For my research, I will be using calcium alginate. So why calcium alginate? I have chosen calcium alginate due to a few reasons. First, it is the most common method of encapsulation found and done in most labs in the world. Next, it is easily prepared and the components are readily available in our laboratories here. Third, it does not have a harmful effect on the encapsulation, on the encapsulated E. coli and also to us. Finally, it is the most cheapest method and the most industrial efficient, hence it is the most practical. For the parameters, for temperature, I have studied the research done by Aldercreutz et al. In 1985. Unfortunately, not much study has been done regarding encapsulated E. coli against temperature. However, the research done by Aldercreutz was done for encapsulated gluconobacter oxidants or G-oxidant cells, and has shown that encapsulated G-oxidant cells can survive up to 50 degrees Celsius, while free-living G-oxidant cells can survive up to only 40 degrees Celsius. E. coli, on the other hand, could survive up to 49 degrees Celsius, but the optimal growth of E. coli would be below 40 degrees Celsius. Hence, I would expect the trend shown by encapsulated G-oxidant cells would be shown by encapsulated E. coli cells during my research. Here is the graph done by Aldercreutz. It shows, as you can see, it shows that for free living cells, As the temperature reaches above 40 degrees Celsius, the relative activity is zero, while immobilized cells continue to thrive until 50 degrees Celsius. For the pH value, I've studied an article by Buzas et al., and the title of his research is Influence of pH on the Growth and Internal Production of Free and Mobilized Saccharomyces cerevisiae cells, or Baker C cells. The free cells of the S. cerevisiae are dependent on pH value with the optimal pH value of 4. However, when it is encapsulated, it can be seen that it is independent of pH value for pH values between 2.5 and 6.2. The free living E. coli cells could survive up to 7.5 pH value since it is neutrophilic. Hence, it can be expected that the same trend would apply to immobilized cells as shown by the S. cerevisiae cells. Here is the graph done by Buzas. As you can see, as for free living cells, as the pH increases above 4, the relative fermentation capacity decreases. While for immobilized Baker C cells, it continues to thrive even as the pH level increases. So for the methods of my research, first I would immobilize the E. coli cells using calcium alginate before allowing it to undergo different experiments, which are viability against extreme temperatures. I will be using 50, 60, and 70 degrees Celsius. Viability against extreme pH level, where I will be using two extremes, both acidic and alkali. For the acidic levels, I will be using 3 and 5. For the alkaline pH extremities, I will be using 10 and 13 pH level. For viability against chemicals, I will be using sodium dodecyl sulfate or SDS. The conditions of my control would be at room temperature 37 degrees Celsius and a pH level of 7. The conditions of my experiments would follow as such unless stated otherwise. For E. coli immobilization, first I will prepare the E. coli as mentioned here. Then I will immobilize. I will produce 20 samples of E. coli before I will immobilize 10 of them. For immobilization using calcium alginate, I will be using sodium alginate and calcium chloride and the E. coli samples. It would take about an hour to produce immobilized E. coli samples from the free living E. coli samples. Then I would undergo the experiment viability against extreme temperatures. 6 samples will be used, 3 encapsulated and 3 non-encapsulated, with the temperatures chosen as mentioned before. For each temperature, there will be 1 encapsulated sample and 1 non-encapsulated sample. The samples will be left in each designated temperature for 48 hours before undergoing CFU or colony forming unit analysis. The temperatures will be controlled by a water bath found in our lab. Next, for the viability against extreme pH level, I will be using a buffer solution to control the pH, which 4 buffer solutions will be made. For the acidic pH level, which is 3 and 5, I will be using an acetate buffer. While for pH level 10, a phosphate buffer, and pH level 13, a potassium chloride and sodium hydroxide buffer. The table below shows my buffer solution, the pH level, and acids and bases I will be using. Next, for the viability against extreme pH level, I will be isolating 8 samples, 4 encapsulated and 4 non-encapsulated. The cells are challenged for 90 minutes at a pH level of 3, then inoculated. Next, the culture will be incubated with shaking at 30 degrees Celsius. This is to shock the cells first. The samples will then be subjected to the 90-minute challenge in each designated pH level before undergoing CFU analysis, as you can see from the graphical representation below. These methods are done by Jordan et al. In 1999. Next, for viability against chemicals, I'll be using methods done by Waldrinch et al. In 1972, in which 4 strains will be used, 2 immobilized cells and 2 free living cells. The cells are left at the control condition, which are room temperature and pH level 7 for 15 minutes before being exposed in the presence of SDS, which will be dissolved and added to the growing culture. After 10 minutes, the samples will undergo centrifugation before undergoing CFU analysis for further research. For my expected outcomes, what I would expect from my research is, I aim to identify the viability of encapsulated E. coli against harsh conditions such as extreme temperature, pH levels, and chemical interaction. Next, the degree of viability of encapsulated E. coli for each variables mentioned before would be determined. The importance of my research are: first, the knowledge from this research can be used in the practical field, particularly in E. coli storage and the industrial field. Next, my research would allow us to understand more about cell encapsulation technology and further improve it for the betterment of biotechnology. Here is my work schedule. My proposal has ended in January and will start my experimental implementation in March before submitting my report in September 2022. Here are my listed references for my proposal and my presentation. And this concludes my presentation for today. I would like to thank you for taking your time in listening to my presentation and wish you all a good day. Thank you. Assalamualaikum warahmatullahi wabarakatuh.